Chapter 26, we're going to be looking at phylogeny and the tree of life. Investigating the tree of life. Take a look at the creature on our screen here. What is it? The obvious answer would seem to be a snake, except it's not. This is a lizard. It's from Australia named scalyfoot, and it happens to be legless. It can be difficult to appropriately classify organisms if we look at just the appearance or the morphology. The scalyfoot does not have a fused eyelid like a snake, doesn't have a highly mobile jaw like a snake, and a short tail behind the anus. Those are all characteristics of snakes, and scalyfoot has none of those. As you might have seen, investigating the tree of life isn't exactly as easy as it appears to be. Phylogeny. This is the evolutionary history of a species or of a group of species. And phylogenies are constructed using systematics. This is the classification of organisms and determining who's related to who and who shares common ancestors. Looking at this family tree, this is a phylogeny, and we are showing in this particular phylogeny that mushrooms are more closely related to us than mushrooms are related to flowers. The closer two organisms are in relation to one another, the closer a common ancestor they will have. And where these branches would be, that would be where a common ancestor would be located. Do we have a common ancestor with plants? Yes, we do, back down here. However, we are more closely related to mushrooms because we have a more common recent ancestor. Classification of those phylogenies, which show those relationships, common ancestry, is a process called taxonomy. Taxonomy was it was popularized by Carolus Linnaeus in what is called the Linnaean classification system. Excuse my spelling. The Linnaean classification system is hierarchical or tiered. And we talked about this briefly. We'll look at an image on the next slide. And it incorporates a naming system for all living things called binomial nomenclature, another thing that we spoke of. So it's assigning each living thing a two-part name using the genus and species taxons. So let's check out that taxonomic tree right here. The genus and species are used to create a name for this particular panther. It's Panthera pardus. We can see the name of him right here, Panthera. That is the name of the genus that he belongs to. Pardus is his specific species name. Binomial nomenclature is putting those two names together to create a scientific name for him. And the nice thing about using this method is this particular language is common across all continents. All continents speak this language. So you don't really have those language barriers in terms of science, so to speak, whereas you do if you're speaking common street tongue. So this shows us the different taxons: domain, kingdom, phylum, class, order, family, genus, and species. The last time we looked at this, it was actually going from the larger ones down, and this time we're looking at the larger ones on the bottom. We're moving up. Either way, it doesn't really matter. We're moving in the same direction, so to speak, towards a more specific group of organisms the closer we get to species. Currently, all life is categorized into one of three domains. And those three domains are called eukarya, archaea, and bacteria. Everything that we know of on our planet fits into one of those three domains. Here we see a phylogenetic tree, and phylogenetic trees help link together classification as well as phylogeny. We can see on this which organisms share higher degrees of relationship than others. we can see that this guy and this guy, they have a common ancestor right here. And this guy and this guy, they have a common ancestor way back here. The tree also shows representation of where those common ancestries is likely to have occurred. There are occurrences where a species is incorrectly placed from time to time. And sometimes we'll think that one organism is more closely related to one organism, a different one, and we're mistaken. Modernized techniques that we're using today, like assessing gene sequences specifically, has allowed for us to make corrections of these mistakes. Once again, sometimes two organisms that appear quite similar are considerably more different than we think, going back to that image we saw in the first slide of Scalyfoot. Now, the Linnaean classification system, with all those taxons we looked at, starting at the top with the 3 domains and working progressively towards the species, it's quite useful. The problem is it doesn't show an evolutionary relationship. That and it isn't exactly going off of gene sequences. It uses a lot of morphological features or anatomy, which looking at Scalyfoot is not the best route. So the more modernized technique is to use something called phylo-code. Phylo-code is a little bit controversial because it would change the way that we pretty much classify everything. It would readjust the way that the taxons are defined. For example, birds and reptiles, they currently belong to different classes. So I'm going to go back to the slide a moment ago. Right here, they belong to different classes. All right, this is a level in the taxonomic tree. The birds belong to Aves, and the reptiles belong to the class Reptilia. However, birds evolved from a group of reptiles. So technically, according to phylo-code, Aves should technically be a subgroup of Reptilia. As you can see, this would adjust a large group of organisms that would need to be entirely reclassified. And that's where the problem comes in. Some people are saying, well, let's just leave it the way it is with this knowledge as being known knowledge, but not actually do it because of the incredible workload that would require. So how do we read a phylogenetic tree? Well, there's a couple points on here that you need to be familiar with. To start with, let's look at a branch point, which is a node. And it's called a branch point because we have a fork, a branch. This represents the divergence of 2 evolutionary lineages from a common ancestor. So here we have one lineage and a second lineage from one common ancestor. Next, we have sister taxa, which are right here. We see B and C are sister taxa. These are groups of organisms that share an immediate common ancestor, and hence they are the closest relatives. So these guys are the closest known relatives to one another because they have a very recent common ancestor. Next, we have one that's not on here called a root point. A root point is usually the furthest thing to the left, which would be right here, that indicates the common ancestor to all organisms that would be found to the right of it. So whatever organism existed right here, that is the common ancestor of all these taxons. Another thing that we need to be aware of that's not actually illustrated on this particular phylogenetic tree is called a basal taxon. B-A-S-A-L, basal taxon. A basal taxon would branch off from the initial ancestor right here, just like these two do. We could draw one in. A basal taxon would branch off right here and continue in this fashion without any other forks in the road. That's a basal taxon. And the last one we need to look at in this would be a polytomy, which would be right here, which is simply where there's a common ancestor and then we have more than 2 taxons derived from it. So poly would be many. We have many taxons from one initial common ancestor. So to infer phylogeny, or evolutionary history, scientists need to look at morphology, once again the anatomy, also genes, and also biochemistry, things like metabolism and other chemical processes. We talked about cellular respiration and photosynthesis as well. So we're going to be checking those out. Morphology, once again, anatomy. We can compare anatomy of various organisms to look for common ancestry. One of the things we looked at previously was homologous structures, or appendages in this case, of different mammals, all the same bones though, just adapted for a unique purpose that's specific to the organism, such as flight or swimming or throwing, running, etc. Not to be confused with analogous structures, which will be discussed in a little bit. The second way was by comparing gene sequences or genes, which tends to tell a more accurate depiction of true ancestry relative to morphological data. The more genes that two different species have in common with one another, the more closely a common ancestor would typically be found. And in some cases, very great similarities in terms of anatomy, implying what would seem to be common ancestry, can actually have very, very small genetic differences. And sometimes very, very small genetic differences can produce two entirely different looking organisms, which can get a little bit confusing when we look at the homology and analogy of it all. Case in point would be the Australian moles versus the North American moles. Very, very similar in appearance. They have an elongated body. They have enlarged front paws, burrowing and digging, small eyes, a thick piece of skin on their nose for protection. However, the internal anatomy of these two guys, the physiology or how their bodies function, the reproductive systems, all very, very, very different. And although these two moles do have a common ancestor, that common ancestor was 140 million years ago, so older than dinosaurs. Their common ancestor wasn't even mole-like, so their common ancestor didn't have these adaptations to pass them down. These had to have been derived at some point in their evolutionary history. What this means is that these front claws or paws and the snouts are there more or less by chance, not because they got them from the same ancestor. So these adaptations, they arose independently of one another as their lineages diverged. Now their similarities today, once again, they're strictly by chance of occurrence as dictated by the changing planet as well as the changing environment. These adaptations then, the front paws, the snout, elongated body, the eyes, those adaptations would be analogous. Analogous because they happen to be similar, but they did not get those similar adaptations from the common ancestor. In order to be homologous, they would have had to have got all those traits from the common ancestor. But instead, these are features that arose independently, but they are the same. Because they are arising independently and they are the same, they are called homoplasies. That is a vocab word that you'd want to know. H-O-M-O-P-L-A-S-I-E-S. So homoplasies are features that are similar, but they're derived from chance, not from common ancestry. We'll pick up with the next slide in the second installment of the video.